Today we're going to be discussing the ear, and we'll talk about the external ear, we'll talk about the middle ear, and we'll talk about the parts of the inner ear. The inner ear we'll look at microscopically. First of all, let's take a look at the outer ear, oftentimes referred to as the auricle. Now this model does not have the tragus. The tragus is a little flap of tissue that of course is oftentimes hairy. The term tragus means goat's beard. Now, sound is going to travel through the external auditory meatus or acoustic meatus, and it's going to make its way through the temporal bone, vibrating a membrane that we're going to get to right about now, and that is the tympanic membrane. The tympanic membrane, of course, vibrates three bones. So sound is traveling through, vibrating the bone called the malleus, which is going to vibrate the incus, which is going to vibrate the stapes. So malleus, incus, and stapes. Now all three of these bones, of course, are going to vibrate together, and they're going to be sending signals to the inner ear. So there's the malleus, there's the incus, and finally the stapes. The stapes is going to be vibrating the oval window going to the inner ear itself. Now the inner ear has a little shape called the cochlea, and that of course is a snail-shaped structure. This is the part where we're registering hearing. We also have the vestibular region, which is for balance, which contains the semicircular canals as well. Now both of these are associated with the vestibular cochlear nerve, and that is the nerve for hearing and balance. Again, the cochlea is for hearing, whereas the vestibular region is for balance. All right, let's take a look at another model. Let's talk about the inner ear. Now, of course, this is the cochlea, which is the part that we use for hearing. And the cochlea is only about the size of, maybe half the size of my fingernail, so it's actually quite small. The other structure that we see here is the vestibular region, and that contains the utricle as well as the saccule. The utricle is used for horizontal balance, and the saccule is used more for vertical balance. Here are the semicircular canals, and at the base of each semicircular canal is an ampulla. The ampulla is where the sensory mechanisms for balance occur, at least for angular balance. You can also see that there's a pink region and a gray region. The pink region is containing perilymph. The gray regions contain endolymph, and we'll talk about this more when we get to the microscopic ear. All right, let's move on to the next microscopic region. This is the microscopic inner ear. One thing we can see are, of course, the three regions that contain perilymph, and endolymph. These are referred to as the scala, and we'll talk about those soon. In this particular image, we can see this nice collection of bipolar cells, which are the spiral ganglion. And the spiral ganglion, let's say that again. This is the region of the spiral ganglion. And these cells are bipolar cells, which are going to communicate directly with the temporal lobe, receiving signals from the sensory mechanisms of the inner ear, which we'll see quite soon. Let's begin, however, with the scala. So this is the scala vestibuli containing perilymph, followed by the scala media containing endolymph, followed by the scala tympani, again containing perilymph. So the scala vestibuli and the scala tympani both contain perilymph. The scala media contains endolymph. So think of PEP, right? Now, this is very rich in sodium ions for the perilymph. The endolymph is rich in potassium ions. It's also rich in positively charged amino acids, making this much more positively charged than making the endolymph much more positively charged than the scala vestibuli and the scala tympani's perilymph. These are separated by membranes, and this first membrane is made of predominantly simple squamous epithelial tissue. That's the vestibular membrane. On the base here we have this broad collagenous sheath called the basilar membrane. This also is going to house the organ of Corti. Notice here this is the spiral limbus. The term limbus just means edge or border. And this is going to support another membrane called the tectorial membrane. Tectorial membrane has hair cells of the organ of Corti embedded within it. It is the deflection of the cells from the tectorial membrane that actually triggers the cochlear part of the vestibular cochlear nerve. In many slides, the tectorial membrane is sprung backwards like this. So it should actually be in this region here. And of course, these are the organ of Corti. Now they contain little cilia, little stereocilia, which actually embed within the tectorial membrane. And again, when the basilar membrane vibrates, we're going to have a mechanical action that triggers the hair cells, which in turn trigger the bipolar cells, which in turn send signals to the temporal lobe, recognizing these pressure waves and mechanical receptors as hearing. Now, if you recall the ampulla was the bulge at the base of the semicircular canals. This is a structure called the crista ampullaris, which bends with the flow of endolymph. So when we're spinning around, the endolymph starts flowing in the semicircular canals, causing in particular this little sail-like structure called the cupula to bend. This in turn is going to fire cells within the crista ampullaris, which are going to be communicating with, among other things, the cerebellum. This gives us our sense of balance. All right, well, that just about wraps up the ear.